So first, thank you for the introduction and thank you for having me here today. I'm going to talk about how transcriptionally active HERV-H Retrotransposons Demarcate TAD domains in human pluripotent stem cells. As you know, at the megabase scale in hexanucleus, we often see these condensed triangular structures at the diagonal, which we often call TADs or loop domains. In mammalian cells, most of these are very likely found by cohesin-mediated loop extrusion that are constrained by CDCF binding sites. Previously, TADs were observed to be largely conserved between different cell types and between related species, and were regarded as stable units in the chromatin organization. It has also been suggested to be instructive for enhancer-promoter communications. So, however, most of the previous Hi-C data sets were relatively low resolution. So in order to study how the TAD structure is related to gene regulation, we generated high-resolution Hi-C here to profile the chromatin conformation dynamics during in vitro cardiomyocyte differentiation. And we wanted to study whether there are any stage-specific TAD boundaries that may be important for cardiac lineage specification. So initially, we observed that the number of TADs decreased during the differentiation, and you can appreciate that the loss of TADs predominantly occur at day 2 and day 80 of differentiation, which we term as ES cell positive TAD boundaries or ventricular cardiomyocyte negative TAD boundaries, respectively. And then we asked what sequences were enriched at those boundaries. Interestingly, we found several classes of repetitive elements were highly enriched in the ES cell positive TAD boundaries, whereas none of them were enriched in the VCM negative TAD boundaries. And among those repeats, among those boundaries, only the TAD boundaries intersecting LTR7 and HERV-H elements shows a very striking ES cell specific expression. So at this point, it looks so interesting that we already started to forget this was a cardiomyocyte related study, because HERV-H is a retrotransposon. It implies that this retrotransposon may introduce new TAD boundaries during evolution. In fact, HERV-H is one of the most successful endogenous retrovirus in the human genome, which contains more than 1,000 copies of HERV-H. And it has been integrated into our genome very recently. And several previous work has shown that HERV-H has been integrated into the human pluripotency network. So genome-wide, among the 1,000 copies of HERV-H, only a few of them are really highly expressed in the human ES cell and other pluripotent stem cells. We found that only the top 50 highly transcribed HERV-Hs are forming TAD boundaries. And also, when these top 50 HERV-Hs are silenced during the differentiation, the TAD boundaries are immediately gone at day 2, which is the mesoderm stage of the differentiation. So these two evidence strongly links the transcription of HERV-H to the TAD boundary strength. And then to first confirm that HERV-H truly are responsible for these ES cell positive TAD boundaries, we deleted 2 different HERV-H sequences in the genome using CRISPR, And in both cases, the TAD boundaries were gone after deletion of HERV-H. This is shown by the change in the contact matrix and also in the change in the directionality index scores. And in addition, we found that when HERV-H were deleted, only the genes on the 5' TAD domain were significantly downregulated, suggesting that HERV-H promoter may act as enhancers, which is consistent with recent reports that HERVs might have an enhancer function. And our data also suggests that HERV-H's enhancer function might be blocked by the boundary so that it cannot activate genes on its 3' end. So in the previous slides, I mentioned that there is a correlation between the HERV-H expression and the TAD boundary strength. So in addition to the knockouts, we investigated whether the HERV-H transcription is also necessary for the boundary formation. So we blocked the transcription of 2 HERV-Hs by the D-Cas9 CRAB system and 2 guide RNAs that targeting the promoters. So you can see here that each guide RNA specifically reduced the targeted HERV-H expression by about 70%, while not affecting the non-targeted HERV-H, and also accompanying the reduction of the transcription, the corresponding TAD boundaries are also weakened if not removed. You can see here in a change in the contact matrix and also in the directionality index scores. So this data suggests that active transcription is necessary for HERV-H to form TAD boundaries. However, it does not distinguish whether the transcriptional activity itself or the HERV-H RNA product is involved in forming the TAD boundary. So to address this question, we further analyzed the solo LTRs in the genome. So solo LTRs are single LTRs, which is the HERV-H promoter. These are LTRs without any HERV-H internal sequences. They were proposed to be resulted from unequal homologous recombination. So we find that solo LTRs, although they express typically at lower levels than full-length HERV-H virus, some of them are also capable of forming the TAD boundaries indicated by the contact matrix and the directionality scores. So this data suggests that HERV-H RNA may not be important for the TAD boundary formation. Instead, the transcriptional activity is more likely the mechanism. So to further explore which candidate proteins are involved in the HERV-H TAD boundary formation, we analyzed the ChIP-seq data from ENCODE and other sources in the pluripotent stem cells. So first, we found enrichment of various forms of POL2 and all 3 subunits of cohesin complex in the highly transcribed HERV-Hs, as well as other active histone marks and some transcription factors. So when we further zoom in at the HERV-H locus, when we plot the ChIP-seq signals in those loci, we observe that HERV-H frequently reads through the 3' end of itself, which is called a 3' LTR. And we also observed broad enrichment of POL2 as well as enrichment of POL2 and cohesin complex. So here is showing the subunit SMC3. And we also observed weak enrichment of CDCF, all of them on the 3' end of HERV-H. So this data suggests that maybe the cohesin complex is positioned to the 3' end of HERV-H by Pol2 when when HERV-H is being transcribed. And this is consistent with several papers reports that cohesin might be positioned by Pol2 in addition to CTCF in mammalian genome. So let me summarize the key observations so far. First, we found HERV-H transcription can create TAD boundaries, and Pol2 and cohesin were found to be enriched at the 3' end of HERV-H. And we also saw that HERV-H can directionally activate genes on its 5' domain. So putting all these evidence together, we think that when HERV-H is being highly transcribed, it forms the TAD boundaries on its 3' end by Pol2 mediated cohesin localization or traffic jam, because Pol2 could be a physical barrier for cohesin. And in addition, the HERV-H promoters may serve as enhancers to activate genes on its 5' domain. However, because of the boundary, it cannot activate genes on its 3' TAD domain. Because, like I said, the HERV-H is a retrotransposon, we further examine the history of HERV-H during the evolution. So HERV-H first invaded the primate genome roughly 50 million years ago, and they greatly expanded the copies in the old world monkey lineage, including humans and human relatives, and to investigate whether HERV-H brings new TAD boundaries into the primate genome, or they preferentially insert into existing TAD boundaries. We perform additional Hi-C on iPS cells from two close relatives, bonobo and chimpanzee, and we also perform Hi-C on a New World monkey species called the marmoset, which contains at least 10 times fewer copies of HERV-H. And we also analyze data from the mouse ES cell, which has no HERV-H in the genome. And consistent with our prediction, so the HERV-H DNA and also its associated TAD boundaries can be observed in the bonobo and the chimpanzee iPS cells, whereas neither the HERV-H nor the TAD boundaries are observed in the regions in marmoset iPS cell and the mouse ES cell in the regions syntenic to human. So these data confirms that HERV-H has introduced new TAD boundaries during the primate evolution. So HERV-H sequences in the human genome are so frequently mutated that they can no longer jump around. To kind of simulate this process, we use the piggyback transposon to help transpose HERV-H into random places in the human genome. So using proximity ligation principle in Hi-C data, we identified 52 insertions in 2 different clones. And not surprisingly, we found that some of the insertions are capable of creating TAD boundaries, like one example that I'm showing here. Whereas in some other cases, the insertion might be silenced and not able to form TAD boundaries. Because piggyback system can only insert HERV-H into one allele of the chromosome, we further performed haplotype phasing and allelic analysis, and we found that in the mutant cell, the alleles with the predicted HERV-H insertion can indeed create contact insulation in the predicted inserted allele. And the panel on the right is showing the combined profiles from all of the loci with the HERV-H insertion, showing that indeed it can create contact circulations in several places. So it further confirmed that HERV-H is capable of creating chromatin domain boundaries when it is inserted into the genome. So, so in summary, we show that HERV-H retrotransposon creates TAD boundaries in the human pluripotent stem cells. We show that deletion or silencing of specific HERV-H elements can eliminate corresponding TAD boundaries, while de novo insertion of HERV-H introduces new TADs. And finally, we show that HERV-H associated TAD boundaries are indeed gained during primate evolution. So with this, I'd like to thank my advisor, Bingren, for his supervision and continued support for this project. I also like to highlight two other co-host authors of this project, Dr. Sebastian Bristol and Dr. Ting Li. Our manuscript is currently available on bioRxiv, and if you're interested in more details, please check this out. And with this, I'm gonna stop and happy to take any questions.